Hi guys, David here with another science update. So the first paper that I want to talk about today is from Nature Communications, and it's very interesting, and it's called Differentially Expressed Genes Reflect Disease-Induced Rather Than Disease-Causing Changes in the Transcriptome. We'll unpack that, don't worry. So the abstract says comparing transcript levels between healthy and diseased individuals allows the identification of differentially expressed genes, which may be causes, consequences, or more mere correlates of the disease under scrutiny. In other words, what they're talking about is if you look at the genetic profile, so the expression of all of the DNA of different individuals, some who are healthy and some who have a specific disease or complex trait, you'll notice differences between those genetics, right? Some will have more of a certain gene expressed and some will have more of a different gene expressed. And so the question is if you note those differences between a healthy person or someone with a specific disease or complex trait, is it that the gene caused the disease? Or perhaps the disease caused the gene to be more expressed? Or is it just a coincidence? And those things are very difficult to untangle. And the present paper proposes a method to basically what's called decompose this correlation. Take it apart so that you can tell if the disease caused the gene or if the gene caused the disease. They do this for a number of different traits by the application of what's called Mendelian Randomization. And in particular, they have an approach that they're calling reverse transcriptome-wide Mendelian Randomization. So to look at a figure from the paper, basically what they're saying is, and I won't talk through all of these acronyms because there's not a whole lot of point, but basically the idea is you have this gene expression and you have it seems to be correlated with some complex trait. But there are confounders present, which is to say there's questions as to whether the causation goes in one direction or the other. So the idea is that you can apply these statistical techniques, these Mendelian randomizations, to try to disentangle whether the gene expression more or less causes the complex trait or if it goes the other way. So looking at figure 2, this is actually the compendium of their application of these statistical approaches to a number of different conditions. They have people with different levels of BMI, different HDL or high density lipoprotein, cholesterol, and then triglyceride levels. And the main thing that I want to point out is for each of these, there is on the y-axis the proportion of correlation explained. And then on the x-axis, so the horizontal line, is the level of the BMI or the HDL or the triglyceride. And blue shows basically the forward correlation, and red shows the reverse correlation. And according to their results, you can see that as the BMI increases, for instance, the red dots go up and the blue dots stay level. Same thing with HDL and same thing with triglycerides. So in other words, for these three complex traits, the results of their attempts to use statistics to disentangle whether the genes cause the disease or the disease cause the genes seems to be weighted towards the complex trait, like the BMI, actually is causing the genes to be expressed differently rather than the other way. And so if this is reliable and if this is true, this may undercut some people's conclusions about whether genes are observed to cause certain diseases or whether the diseases are just simply causing certain genes to be overexpressed. Like that would look like, for instance, if you were obese, maybe you have system-wide inflammation and that's causing genes that are correlated with system-wide inflammation to be overexpressed, rather than having genes that cause system-wide inflammation increasing your likelihood to be obese. So something that's always important to point out is the limitations. So they say our approach has its limitations which need to be considered when interpreting results. They say, first, our results are mainly focused on gene expression levels in whole blood. They say this is primarily due to the reduced power resulting from the small sample sizes when conducting tissue-specific analyses. However, because gene regulation is tissue-specific and many diseases manifest themselves only in certain tissues, future possibilities to interrogate larger and more diverse tissue-specific datasets could unravel causative disease links for genes not differentially expressed in blood. In other words, they're just looking at the genes in the blood, but they're saying, well, maybe, for instance, if we're talking about BMI, we should be looking at the gene expression in adipose tissue or fat tissue, and maybe that would give a different result. So, in other words, they're not really ruling out the possibility that gene expression is causing these complex traits. They're really just showing that as far as their current data set allows, it seems like you can't really make the inference that the genes are causing the complex trait or the disease. If anything, it seems to go the other way, but it's very preliminary data. They say another caveat lies in the fact that differences in power makes it difficult to compare the results of these two different statistical approaches, the forward and the reverse statistical approach. One of the most important determinants of statistical power for Mendelian randomization is the sample size available for the outcome. Thus, the reverse Mendelian randomization approach is less powered, picking up mostly strong effects. So in other words, their math for the forward causation versus the reverse causation is the math for those two different processes are basically going to be different because of the sample size associated with the two processes, and they say that will undermine the accuracy of the results. But overall, as preliminary data, it's pretty interesting. They say, in conclusion, our bidirectional analysis disentangles the causes and consequences of gene expression for complex traits and reveals that complex traits have a more pronounced impact on gene expression than the reverse. The next article that I want to read today is called Even Mild Cases of COVID May Leave a Mark on the Brain. And this is interesting. This is from a neuroscientist, a cognitive neuroscientist. And she says, in August 2021, a preliminary but large-scale study investigated brain changes in people who had experienced COVID-19 drew a great deal of attention within the neuroscience community. In that study, researchers relied on an existing database called the UK Biobank, which contains brain imaging data from over 45,000 people in the UK going back to 2014. This means crucially that there was a baseline data and brain imaging of all those people from before the pandemic. So this is a best case scenario from a scientific point of view. You know, you want to find out, for instance, if COVID-19 caused there to be changes in brains. Well, to really make that connection, you would have to have a really good sense of what people's brains looked like before COVID-19. But thanks to this database, they basically have that. They have a baseline to compare changes to. So the research team analyzed the brain imaging data and then brought back those who had been diagnosed with COVID-19 for more brain scans. They compared people who had experienced COVID-19 to participants who had not, carefully matching the groups based on age, sex, baseline test data, and study location, as well as common risk factors for disease, such as health variables and socioeconomic status. The team found marked differences in gray matter, which is made up of the cell bodies of neurons that process information in the brain, between those who had been infected with COVID-19 and those who had not. Specifically, the thickness of the gray matter tissue in brain regions known as the frontal and temporal lobes was reduced in the COVID-19 group, differing from the typical pattern seen in the group that hadn't experienced COVID-19. What's very interesting is that when the researchers separated the individuals who had had severe enough illness to require hospitalization, the results were the same as for those who had experienced milder COVID-19. So that's interesting. So this is a study where they have a really good baseline, and they're saying that it looks like those who had COVID-19, whether it was a mild case or a severe case, had noticeable loss of brain tissue. Now, this is rather upsetting. They say, finally, researchers also investigated changes in performance on cognitive tasks and found that those who had contracted COVID-19 were slower in processing information relative to those who had not. So in other words, it looks like not only did the COVID-19 patients, whether they were mild cases or severe cases, have noticeable brain tissue loss, but they were also cognitively slower. Like they had slowed down a little bit compared to people who had not had COVID-19. We don't know what happens, for instance, to a COVID-19 patient 5 years later. Maybe they recover 100%, and I really hope that's the case. But it looks like a lot of people have brain tissue loss at this point, even people who had mild cases of COVID-19. And that's very interesting from a scientific point of view, but it's very upsetting from a humanitarian point of view. So that's really sad. So on to something less depressing for our last paper. This says, rejuvenated InSight hears 3 strong Mars quakes. And it says NASA's InSight Mars lander has hit 2 major milestones: 1,000 Mars days on the surface and the detection of the 3 largest Mars quakes ever recorded. What's actually the thing that I want to talk about is not so much the earthquakes, but this interesting thing that they did to rejuvenate the Mars InSight Mars lander. So what happened is that the Mars lander has solar panels, obviously, to power it, but it had accumulated a lot of Mars dust. And this dust had reduced the capacity of the lander's solar panels by nearly 80%. Sometimes with these kind of things, you'll have breezes come by to clean off the solar panels. And that actually has happened before to the Spirit and Opportunity rovers in the 2000s. But without a strong enough wind, which didn't seem to be coming by the region where this particular lander was, the dust clings pretty well. So they came up with this idea of creating a whisk broom effect. And obviously they can't just like make a broom or something and send it to Mars and clean off the rover. They can't do anything like that. So they have really like limited tools. So they do have the lander's robotic arm. And what they decided to do is try to put sand on the upwind side of one of the solar panels and let the wind whisk it across the surface, knocking some of the dust free in the process. The idea, Bannert admits, sounded so preposterous that when the scientists first suggested it, they pretty much got laughed out of the room. The last thing you want to do is put more dirt on the panels when they're getting dirty. But still, everyone had seen images of how blowing sand had removed dust on the Martian surface, so with no better option they tried it. And it worked. It didn't sweep away all the dust, but it cleaned away enough that the lander is again very much a going concern. So they were able to clear off the solar panels, and because of that, they were able to continue to measure data, which is very cool. So thanks again for watching. I hope you enjoyed these articles and found them intriguing. I hope you will join me again in the future soon, and please consider dropping a like or subscribing. Bye.